In this model, we will be demonstrating the details of meiosis and how Mr. Adone's and Mrs. Adone's chromosomes join together to create Lorenzo. In the first step of meiosis, there's interphase. As you can see, there are duplicated centrosomes and the DNA is extremely loose. It is not tightly bound up. However, as we go through interphase, the DNA begins to tighten and condense. The DNA is also duplicated. In prophase 1, the chromosomes are still being condensed and getting packaged, and the nuclear membrane disappears. The blue chromosomes represent the paternal chromosomes, which are from Mr. Adone, and the purple chromosomes represent the maternal chromosomes, which are from Mrs. Adone. However, Mrs. Adone carries a mutated AOD gene on one of her chromosomes as represented by this bead. After each duplicated/replicated chromosome pairs with its corresponding homologous chromosome, which are chromosomes that are extremely similar, they undergo crossing over. Crossing over is when sections of the chromatids cross over each other, and these cross sections of chromatids, which contain alleles, are exchanged. During metaphase 1, paired homologous chromosomes line up in the middle of the cell. During metaphase 1, independent assortment occurs. Independent assortment is when the homologous chromosomes line up in the center of the cell, but the order in which they are placed is random. They could have easily been organized like this or like this. The centrosomes move to either side of the cell, and the spindle fiber from the centrosomes attaches to the centromere of the chromosome. In anaphase, the homologous pairs of chromosomes separate. Spindle fibers pull each homologous chromosome towards opposite ends of the cell. During telophase/kinesis, a nuclear membrane forms around each cluster of chromosomes. The cytoplasm divides, and the rest of the cell is divided to create two new cells, finishing meiosis 1. In prophase 2, the nuclear membrane disappears as the chromosomes begin to condense again. The centrosomes begin to appear as well. After prophase 2, metaphase occurs. The centrosomes begin to line up in the center of the cell. During anaphase 2, spindle fibers from the centrosomes make the two sister chromatids separate, and the individual chromosomes now move to opposite ends of the cell. The double-stranded chromosomes are split, now becoming single-stranded. At the end of telophase/kinesis, 4 haploid cells are formed which receive 2 chromosomes each. A nuclear membrane forms around the chromosomes. These haploid cells will turn into sperm or egg cells, which are a type of gamete. When one egg cell unites with a sperm cell, fertilization happens. This forms a zygote, which will eventually form into a baby. Since AOD is a sex-linked disorder, when Lorenzo received his mom's chromosome, he also received the affected gene that contained AOD. If Lorenzo were female and his dad gave him an X chromosome, he would be fine, since the other X chromosome would cancel out the defective one. However, since he received a Y chromosome, he was diagnosed with AOD.
Lorenzo gets 50% of his mom's DNA and 50% of his father's DNA during metaphase 1. When homologous chromosomes line up double file, then it goes through multiple other steps until cytokinesis, when there are 4 haploid daughter cells are formed, each having only 1 chromosome of each homologous pair, which means that both of Lorenzo's parents, his mom and his dad, both of them combined equal 4, and so that's what he gets. And so some of both of their DNA is in some of these daughter cells. Meiosis brings about genetic variation during prophase of meiosis 1. The double chromatid homologous pairs of chromosomes cross over with each other and often exchange chromosome segments. When they recombine like this, it creates genetic diversity by permitting genes from both parents to intermix, creating chromosomes with a different genetic variation.